Hello, this is Oluwanifemi Adekanbi. I'm Femi from Group C and I'm presenting my individual project presentation on enzymes. What are enzymes? Enzymes are proteins that serve as biological catalysts to speed up chemical reactions. They speed up chemical reactions by lowering the activation energy level using a catalyst. They do this by inducing a conformational change in substrate which weakens chemical bond. Enzymes are important because they speed up the reaction and regulation reaction rate. Now, what is trypsin? Trypsin are proteins, which are enzymes that break down protein. Trypsin catalyzes the reaction of protein hydrolysis, which are translocated in trypsin's active site. They digest the dietary protein in the small intestines of humans. Trypsin is active in the small intestine of humans, and they typically have a pH level of 8. In bacteria, trypsin is secreted from the bacterial cell, and the pH, temperature, and salinity varies. And our enzyme question is, what is the activity level of enzyme trypsin at 3 different temperatures, 45, 55, and 65 degrees? Our hypothesis was the activity level for trypsin will be the highest at 55 degrees. And our rationale was, in our first experiment, we tested the activity levels at 25 degrees, 50 degrees, and 75 degrees. The activity level was the highest at 50 degrees. We wanted to test the different temperatures closer to 50 degrees to find what the optimal temperature would be in a smaller range, since 50 degrees had the highest activity rate. So we expected 55 degrees in the range between 45 and 65 to have the highest activity levels. For our materials and method, our experimental variable, we tested temperatures at 45, 55, and 65 degrees. Our control variable was pH, pH was at 8, and our negative control was DH20, which was distilled water. And for replicates, we had 5 replicates and 3 negative controls for each temperature. So azocasin was our substrate, and azocasin is modified milk which contains sulfanilamide that is attached randomly, and it allows the enzyme to be adsorbed at 440 nanometers. We had 3 experimental temperatures which we labeled A, B, and C. We have 5 replicates of each variable, and we also added 3 controls for each temperature we used. And we used replicates because they provide more accurate data, they identify outliers, and they measure variability. So like I said earlier, we labeled 3 sets of 5 tubes. We labeled them A, B, and C, and since we have 5 replicates, they were A, B, C, 1 to 5. Next, we gave each set of tubes 3 control tubes which would not receive trypsin, and we labeled them AC, BC, and CC 1 to 3. We pipetted 450 microliters of our reaction buffer into all 24 tubes, followed by 500 microliters of azocasin into all the tubes. We then added 50 microliters of trypsin in the A to C tubes and closed all 15 tubes. Next, we added 50 microliters of DH20 into all 9 control tubes A, C, B, C, and CC 1 to 3. Next, all set tubes were incubated at the assigned temperature for 20 minutes. Group A and AC at 45 degrees, B and BC at 55 degrees, and Group C and CC at 65 degrees. After incubation, 50 microliters of 10% TCA was added to every tube, and we used TCA to precipitate the undigested substrate. We then centrifuged the reaction after adding the TCA in order to remove undigested substrates. All 24 tubes were then centrifuged for 2 minutes at 12,000 rpm. The supernatant was transferred from all tubes into cuvettes, and the spectrometer was blanked using a cuvette with DH20. Each cuvette absorbance was read and recorded at 440 nanometers. and this was our result. So we have listed 3 trial temperatures, which are 45 degrees, 55 and 65 degrees, and we have 5 experimental values for each of the trials. And after measuring it, we found the experimental average for both the trial and the control, and we found that the average for trial at 45 degrees for our experimental trial was 1.823, for 55 1.782, and for 65 1.758. We also found the control average for 45 degrees was 1.897, for 55 it was 1.61, and for 65 it was 1.747. We then did the standard deviation for each temperature and control to show the extent of deviation for each degree. The standard deviation for the experimentals for 45 degrees was 0.14136, for 55 degrees was 0.03545, for 65 degrees it was 0.1465, and then we also did the same standard deviation for the control, and at 45 degrees we had 0.15621, at 55 degrees we had 0.90852, and at 65 degrees we had 0.23928. Next, we use the t-test to compare the average and standard deviation of two samples to determine if there is a significant difference between them, and the t-test was used to prove the null hypothesis. And the null hypothesis states that when the enzyme activity is above 0.5 degrees, it is considered to not be significantly different, whereas when it's below 0.5 degrees, it is going to be very different. So the results indicated that for whenever the enzyme was at 45 degrees, it is not significantly different at 55 and 65 degrees, whereas for the enzyme activity for control, when it's at 45 degrees at the highest, it is significantly different from the activity at 55 and 65 degrees. The result of the experiment did not support our hypothesis. We hypothesized that the optimal temperature would be 55 degrees. At 55 and 65 degrees, the activities overlapped, and since they are not significantly different as we tested in our null hypothesis, our hypothesis that 55 degrees would be that 55 would be the highest activity level was not supported. We hypothesized that the optimal temperature would be 55 degrees. At 55 and 65 they overlapped. In the future, we would increase the numbers of replicants to make more accurately identify outliers in temperature. We would increase incubation time from 20 minutes to 30 minutes. We could test at small intervals between each temperature time temperatures to find a more accurate optimal temperature. If possible, the temperatures would be at 42 degrees, 45 degrees, and 47, so as to give ourselves a gap of differences to see what the optimal temperature could possibly be. The experimental design and execution was not adequate enough to test the hypothesis because it did not give us enough variety of temperatures to test, so we can come up with different outcomes in order to find the optimal temperature. We concluded that the result does not support the hypothesis with rationale because all 3 temperatures were not significantly different, making them all to be optimal, as opposed to our hypothesis that said 55 degrees would be optimal.